Thanks, Alex, for the kind introduction and also for the invitation to share our work that we have recently posted on bioRxiv, describing how Nodal and Wnt control the emergence of the mammalian body plan. So that was the question that I set to tackle during my postdoc, with whether signaling was involved in establishing the mammalian body plan, and particularly in the development of these different modules that form the body plan. And if you are interested in that, I would suggest you to read this short comment that I made with Alfonso Martinez Arias about this modularity and how that is achieved and what we know from the mouse embryo. And this partnership with Alfonso happened because Alfonso's lab has, as you probably all know, developed the gastruloid model system, and the one you are seeing here is the mouse one, where ES cells, around 300 ES single cell ES cells, are allowed to self-organize, to aggregate and self-organize, and they can mimic development. Particularly, they can recapitulate some features of gastrulation and later on axial elongation. And this happens because of a reason, and that has to do with this treatment that is given between 48 and 72 hours. It's a chiron treatment, so chiron is an agonist of Wnt signaling, and my idea was to try to understand the role of Wnt by using this chiron treatment as a proxy. And for that, I used different amounts of chiron and also played with the duration of this treatment. And what you see here in this principal component analysis is samples, bulk RNA-seq samples of pools of gastruloids that collected at different time points. And as you can see, things change a lot during these early days. For instance, here you see the transition from 0 to 48 hours, which is a transition from naive to formative prime pluripotency. At this stage, at 48 hours, the gastruloids resemble sort of an E6.0 mouse embryo ready to start gastrulation. And then we played, as I mentioned, we played with the chiron treatments, and as you can see here, and it goes from the pink to red in terms of concentration. As you see, when we increase the concentration of chiron, we start to see more posterior related gene markers such as brachyury, CDX2, CDX1. And when I mean posterior, I also mean later markers, as you can see here, for instance, the OX genes and also Wnt3A coming up. This is a different PCA where the same where some of the bulk samples are also plotted, the gastruloid ones at the bottom, and also we also plotted the P1 Sala data set, so the mouse embryos spanning from E6.5 to E8.5, and then you can see here that they are organized along dimension 1 in terms of time, and the same happens with our gastruloid samples. So again, you see with gastruloids that were treated with 0.5 micromolar chiron, they more or less resemble an E7.0 mouse embryo, and with higher levels of chiron, such as 20 micromolar, you see again this appearance of later OX genes and also the CDX genes, and this resembles basically the emergence of the caudal epiblast. You can have a look at our preprint, look at the different marker genes and the heat maps, but basically what we see is a downregulation of pluripotency, a downregulation of the genes that are expressed in the early epiblast and the early primitive streak, here highlighted in green, and then an upregulation with the chiron treatment in a concentration-dependent manner of genes that are associated with the late primitive streak and also the caudal epiblast with axial progenitors. And one of the things that we do not see, and it is interesting, is that despite all these differences, we do not see a significant increase in the amount of mesoderm. So what this suggests is that instead of Wnt being a driver for a specific fate, in this case mesoderm, what we suggest is that Wnt regulates a cell state transition. And this cell state transition drives pluripotency to multipotency, basically drives gastrulation to the formation of the caudal epiblast, which then will continue driving axial elongation. And this happens, as I showed you, in a concentration and a time-dependent manner, and it is because Wnt activates this what we call late posterior primitive streak program, which is driven particularly by brachyury, CDX2, Wnt3A, and the OX genes. And so in this way, it seems that Wnt controls the pace of gastrulation, that Wnt controls developmental time, and this is important. So basically this is a summary of what I showed you. So in red, it is the region of the embryo that is activated by the chiron treatment. And this also explains why the chiron treated gastruloids usually have more posterior body structures highlighted in red and in an E8.5 mouse embryo. But it also raises the question of what could be the regulators of the early primitive streak, and particularly what would drive the formation of these more anterior mesendodermal body structures, and whether we could also mimic these in vitro with gastruloids or not. And so as a prime candidate, we looked at nodal signaling because we know that nodal is highly expressed and highly active at the beginning of gastrulation, particularly in the tissues that we would be interested in seeing. So the rationale was very simple. We switch we replace chiron by activin A, which is an agonist of nodal signaling. With this, the morphology of the gastruloid changes and also their gene expression. So as you can see at 72 hours, the gastruloids, in contrast with the chiron-treated ones, have display low levels of brachyury, but also expression of mesodermin. And this co-expression of these two markers is indicative of the early primitive streak. You can also have a look at the fates that usually arise from the early primitive streak, some at later time points, such as 96 hours in the chiron, with endoderm rising, also noto expression in these gastruloids, and not in the chiron treated ones. And so here what you see in this PCA is basically samples from chiron treated gastruloids and also activin ones. I want to point out here with the 72 hours activin gastruloids in light blue, and also they are more similar to the low chiron treated gastruloids. So this fits with our theory that Wnt is indeed controlling this activation of the posterior primitive streak. And as you can see also here, the activin treated gastruloids follow a different developmental trajectory in blue at the top than the chiron treated ones in red at the bottom. So they are different. And just to give you a few examples, so as you can see here, by the staining of FOXA2 and SOX17, you can see a well-defined gut-like structure in these activin treated gastruloids at 144 hours. We also did single-cell RNA-seq, and you see that in these clusters, you see elements of the precaudal plate and also the foregut with the expression of very anterior markers like LHX1, HSX1, and also DKK1. So we are talking about very anterior regions of the gut. And if we look at, for instance, the notochord, which is highlighted here by FOXA2 and also brachyury, this structure does not contain much noto expression, and that was also seen in the single-cell RNA-seq. So what this suggests is that, again, the notochord-like structure that we are making is very anterior, because these ones are known to lack noto expression, which is only found at the posterior part. So basically, to sum up this second part, we found that indeed nodal is important during early gastrulation, particularly for the early primitive streak, and that it is important in the formation of these structures, these mesendodermal structures, and that we can mimic their development with activin gastruloids. So the question now was, because we knew that Wnt signaling was important, that the gradient was important in this establishment of the posterior part, whether the activity of nodal, which we know decays throughout gastrulation, would also play a role in the development of these anterior body structures. And for that, we did the same, we used a similar protocol, but reduced the levels of activin A. As you can see here, highlighted in red are these gastruloids. They are different from the high activin treated ones. But the key experiment that I want to show you that captures the whole thing is basically this one at the right. So we use the P1-SAL dataset, and we reconstructed different cell populations spanning anterior and posterior expression. And as you can see here, the chiron-treated gastruloids in green cluster more close to the posterior part, whereas the activin treated gastruloids, the high ones, are more anterior, and the low activin ones slightly more posterior. So what this indeed suggests is that this gradient of nodal that occurs during gastrulation is important to define the anterior body formation, and that this is important not only in the concentration but also in a time-dependent manner. You can look at our preprint where we look also at this issue of time. And so differently from Wnt signaling, what we found is that indeed nodal affects phase specification. Nodal tackles a cell state transition, nodal affects phase specification, and with different nodal concentrations generating different mesendodermal body structures. And the way nodal does this is by activating this early primitive streak program where iomesodermin and also FOXA2 play a critical role. So what our paper shows the novelty is in the fact that in the literature, nodal and Wnt are thought to cooperate to build gastrulation, to build a primitive streak. However, what we suggest is that despite this collaboration, there's also an antagonist interaction between the two. And this antagonist interaction is important also in the context of their dynamics, because we know that we start early gastrulation with high levels of nodal and low Wnt, and we go to a high Wnt and low nodal. And it is this antagonist interaction between the two that indeed will make the different body structures. And so this would be a sum of our findings and how we explain this issue of modularity and how the signalings contribute to this modularity. And what we are doing now is playing with specific roles to try to build specific fates in vitro, and also to play with this modularity with chimeras. So to finish, I just want to call your attention to a paper that we wrote also about this modular organization and how it is related with the Spemann-Mangold organizer, and also to a preprint from the lab of Sebastian Arnold that is looking at the same question, but from the point of view of the transcription factors, brachyury and also iomesodermin, and it reached at similar conclusions than ours. And with this, I want to end and thank you, the lab, Alfonso, also Tina, and I want to highlight Pau and Gabriel that helped with the bioinformatic analysis and also the funding agents. And thank you for listening.
Great. Thank you very much, Andre. We'll give people a chance to type some questions into the Q&A box. And in the meantime, I'll start with a question. So I'm sorry if I missed this, but you mentioned that Wnt signaling is necessary for this elongation process. What is the source of the Wnt signaling in the embryo itself?
What is the source of Wnt
Which
signaling?
cells are expressing Wnt to
So the
activate
cells
faster
that
in the embryo?
express Wnt signaling are the ones in the posterior part. This Wnt signaling is supposedly triggered by BMP, which comes from the extraembryonic tissues. This would be the main source.
Okay, and you mentioned at the end that there's this antagonism between nodal and Wnt. At what level do you think that's happening? Is that at a transcriptomic level, or do you think there are shared factors within the signaling cascade that are sort of competing for one pathway versus the other?
I think, and as Sebastian Arnold shows, most of this comes from, then it is run by the transcription factors. So I think what happens is you have a set of transcription factors that are upregulated by this thing, that are triggered by this signaling, and then it will be their interactions that will build this different and antagonistic interaction.
And in your system, is it possible to create a gradient of each of these molecules? Is it dependent on tissue thickness or perhaps applying the signals at one end of the dish versus the other? Can you create a gradient with nodal at one end and Wnt at the other?
In our system, it's not easy, but if you use, for instance, optogenetics or some sort of microchips where you can control the orientation and the presence of these signalings. However, what we are doing now is using chimeras. So we are creating different aggregates with different levels of sync, so you can create these gradients as well. Yeah.
Great, thank you. We have a message in the Q&A box that says, thank you, it's a very interesting talk. If we don't, we have another question that I will ask now, and then we can move on. So, Spiakoza, sorry if I pronounced that wrong, is this a matter of just nodal and Wnt, or do you think there's other factors, morphogens involved?
So there are also other factors. I think Wnt and nodal play a critical role, but there's also the lab of Ram Sambasivan in India is also looking at retinoic acid, and he's seeing that retinoic acid helps this role of Wnt signaling. Basically, it gates what we see that the effects that Wnt signaling is propagating in the cells. So yes, of course, there are other signalings. However, Wnt and nodal play at the very top.
Great. And we have one last question, so I'll ask it. Guillaume is asking, have you looked at the endogenous nodal expression and whether that varies with the concentration of chiron?
So yes, we looked at the nodal concentration, so you can look at the paper as well, and yes, it changes. It changes. Yes.
Great. Thank you very much.